Now on to our next speaker, who is Shichun Gu from University of Bristol.
Hello, I'm Shichun, and I'm going to talk about the last open reading frame of 10 on the SARS-CoV-2 genome. So this talk is divided into 4 different parts. The first is a quick introduction about ORF10, the result we get back from cell cultures and animal studies. The second part will be about 3 different lung models we've used in laboratories. And the third and fourth part were 2 different approaches I'm using at the moment, trying to study further the pathogenicity and the possible functions involved ORF10. So ORF10 is highly structured and conserved. It only presents in SARS-CoV-2 genome, not in the previous MERS-CoV or SARS-CoV-1. Among the 40 million of SARS-CoV-2 sequence held by GASAID, there is only less than 1% of them had a truncated ORF10 sequence. Based on the Wuhan D614G, the image at the bottom is a predicted RNA structure of the ORF10 region. If we mutate both the start and the internal AUG site to a stop codon, and using reverse genetics, we can produce an ORF10 knockout virus. When infecting those AAT cells, which is an adenocarcinoma human alveolar basal epithelial cells using either the knockout virus or the wild-type virus, we don't see a significant difference at 18 hours post-infection based on the host cell quantitative proteomics, but we do see some difference at the transcriptomic level. Theoretically, the knockout virus should not produce any ORF10 proteins because we have switched off the start codon. So I designed a lentivirus which codes for the SARS-CoV-2 ORF10 sequence. And before I infect the cells with SARS-CoV-2, I infect them with the specific lentivirus. And the ORF10 protein produced from the lentivirus should somehow compensate for the loss of ORF10 when I infect those with the knockout virus. So I move on to do some competition assays. The knockout virus and the wild-type virus was mixed in 50-50 at the MOI of 0.01, and then this virus stock was taken and marked as P0. Those P0 were then used to infect 2 stable cell lines. The GFP cell line is those lentivirus, so the VTN cells infected with lentivirus coded with green fluorescent protein only. So this is a control group. The ORF10 condition is the cells infected with lentivirus coded with GFP plus ORF10 sequence. And after the P0 was used for infections, 48 hours after that, the P1 supernatant was collected, and the viral load in P1 supernatant was assessed using quantitative PCR to dilute it back for the MOI of 0.01 and then used for the next round of infection. The infection was repeated 3 times. That's why we have 4 different Sanger sequencing results here. In short, there is no difference when I compare the ORF10 group to the GFP control group. So I wanted to see if I can generate a more saturated version of those stable cell lines. Based on the ORF10 stable cell lines I generated previously, I infected those cell lines again with the same lentivirus. At this time, the plus condition is where I leave the inoculum on during SARS-CoV-2 infection, and the sequential one is I wash the supernatant off before SARS-CoV-2 infection. The conclusion is that the wild type always wins in these specific cell lines. So these were based using a VTN cells. These cells is a Vero E6 cell line with a defected innate immune responses. Whether I infected with control GFP ones or those ORF10 coded lentivirus, the result is always that the wild type replicate much better than the knockout virus. Now move on to using A549A2T2 cells. The study design is slightly different here. I split the cells at the same time and leave a group of the cells untreated, while I treat the other group with universal type 1 interferon 18 hours prior to the SARS-CoV-2 infection. And as we can see at the P0, there is a mixture of knockout virus and the wild-type virus. And in the untreated group, both viruses still replicate to a very similar level, whereas in the universal type 1 interferon treated group, the wild type replicates to a higher level comparing to the knockout group. So in these AAT cell lines, the wild type only wins when we pre-treat these with universal type 1 interference. Despite there is no significant difference when we grow wild type and knockout virus separately and assess their growth on growth curves within 48 hours, in those competition assays, we see that wild type virus wins where there is no innate immune responses, those VTN cells. And in normal innate immune responses cells, those AAT cells, both virus grows to a very comparable level. However, if we pre-trigger those innate responses using type 1 interference, 18 hours prior to the SARS-CoV-2 infection, the knockout virus start to struggle again. So let's look at the animal studies. If the hamsters were infected with the knockout virus or the wild-type viruses, we see a very obvious knockout attenuation. So the knockout virus is highly attenuated in infected hamster lung tissues. There is a tenfold difference, but this difference is not seen in nasal samples. Since we haven't seen such big gap in our previous cell culture models, we wanted to introduce some in vitro lung models in the laboratory. The first one is using primary pneumocyte. So those pneumocytes consist of two different cell types, alveolar type 1 and alveolar type 2 cells. Those pneumocytes will grow in a monolayer, just as the rest of the carcinogen cell lines, and the experiment was done in 6 well plates. The second model is called air-liquid interface, where we have alveolar epithelial cells growing on top of the membrane and the endothelial cells growing at the bottom of the membrane in contact with the medium. There are 3 different groups. The MOG is the uninfected group, the wild-type infected group, or the knockout infected group. Those viral load were added on top of the membrane in contact with the alveolar epithelium for an hour and then washed off. Cell supernatant were taken at several different time points, and the total RNA extraction was done after 48 hours of infection. However, we don't see any significant difference in those 2 models. Then we move on to the third model, which is the induced pluripotent stem cell-derived human lung organelle models. So these cells were treated with specific chemicals, so they can differentiate it into the desired lung organelle structure. Along the pathway at several time points, we would assess differentiation using different phenotypic markers shown in the red boxes here. After 2 months of growth, it will form a spherical airway-like structures, and we would infect those organelles for 4 days. The final replication of SARS-CoV-2 was assessed based on viral nucleocapsid protein expression using immunofluorescence staining or qPCR result. As expected, the wild type replicate as normal, but we can't detect any knockout virus replication in those organelles, which confirms with the animal study. In order to further study the possible functions and pathways involved ORF10, we started to work with a biochemical group in China. So the hypothesis is that the ORF10 peptide would bind to the CYG11B binding pocket on the CUL2 protein, and this would increase the ubiquitination of IFID46, because IFID46 also binds to another binding pocket called C2 on the same CUL2 protein. And the ubiquitination of IFID46 will eventually lead to a higher grade of degradation. If we the essential binding site of this ORF10 protein is the first and the second amino acid. If we assume the wild type had the binding affinity of 1 and a single change of amino acid, for example, if we change the glycine to the pyrroline at amino acid site 1 or change the tyrosine to alanine at amino acid position 2, it will nearly abolish its binding affinity. Since I just started this sub-project, so I only have very limited result to show here. The Western blot shows that the degradation of IFID46 only presents in the wild-type peptide group, not in the G1P or Y2A modified peptide group. On the other hand, I'm also doing reverse genetic. So previous attempts trying to replace the whole ORF10 region with either M neon green or M scarlet reporter protein had failed, while we tried to limit the change to its RNA structures, knockout virus can be rescued successfully. Alongside with those knockout virus, I also designed a synonymous change of 10 sequence. Well, I fully disrupt its RNA structure, but it should produce the same protein. This modification is lethal to the virus, and this cannot be rescued. However, if we pay attention to the hypovariable region right at the end of the ORF10 sequence, and when I try to embed a 6-histine tag or a flag tag at the hypovariable region, this can be tolerant by the virus, and both tagged virus were rescued successfully. At the moment, I have already harvested a lot of samples infected with both tagged virus, and I'm waiting for the result from direct RNA sequencing and transcriptomics. Hopefully, we'll get more confirmed results showing the pathway involved by ORF10. Finally, I would like to thank the Bristol team and the Liverpool team and the fundings. Happy to take any questions. Thank you.
Great talk, thank you. Has anyone got any questions?
Thank you very much. I was very interested by the temperature differences. I was interested by the temperature difference, potentially like upper airway versus lower airway in the hamster. If you try to change your cell culture models at low temperature versus high temperature, could you maybe find that phenotype?
We actually done the temperature sensitive experiment, but I did not include the result here. So when we do the growth curve, we normally do it at 37 degrees Celsius, but obviously we would normally assume the upper respiratory tract is around 37 degrees Celsius. So after we got the result from hamster studies, we set up two different conditions, with one incubator at 33 and the other incubator at 37 degrees Celsius, and grow both the knockout virus and the wild-type virus at two separate conditions. But there is no significant difference caused by the temperature change, so we'll kind of eliminate the possibility that it is the temperature causing the different replication rate in the upper and lower respiratory
What about
tract.
higher temperature?
Higher temperature,
Yeah.
do you mean like 40 degrees
What is
Celsius?
the body temperature of a hamster? Is it 37? It's higher, I think.
Well, we haven't tried 40 degrees Celsius.
Back to the lab. Okay.
I mean, the other question is, do hamsters get a fever response? James?
Thank you. I was wondering if you basically made a temperature sensitive UTR mutant.
No.
Start changing the codon slightly, maybe higher or lower temperature. Oh, it's really cool. Thank you.
Mm.
Another question over here.
So your data showed that your ORF10 knockout mutant could infect and replicate in Calu3 cells, I think it was. Is that right? Calu3?
So it's attenuated in long organoids. Yeah.
But in that same graph, it showed that
it's
would still
slightly lower
replicate in
at
Calu3s. Do you have any idea as to why the knockout mutant can replicate in Calu3 cells, but was absent in organ?
In Calu3, knockout virus can replicate, but to a slightly lower rate at the first 48 hours. But if we keep culturing it for over a week, for example, it will grow to the same level as the wild-type virus. And I would assume because the organoid would trigger more innate immune responses, and based on the previous cell culture result, the pre-triggered innate immune responses, or maybe a higher rate of innate immune responses, will make the knockout virus struggle a lot.
Were you micro-injecting the organoids, or how was your route of delivery?
Do you mean how to infect?
Yeah.
So so after 2 months of growth, it will form like a spherical structures, and 12 hours before the SARS-CoV-2 infection, we would slightly pipette up and down to disrupt its spherical structure so that it can in contact with the SARS-CoV-2 virus. Otherwise, it won't get infected.
Cool. 1 or 2 more questions.
A quick question from me. So ORF10, what is it? Is it an RNA structure that happens to have an AUG and a TRS, or is it a protein?
So that's the question. Like at the beginning of the stories, we're trying to figure out whether it's the RNA structure or it's the actual ORF10 protein that is made this specific sequence essential for SARS-CoV-2. Otherwise, it won't preserve during all those evolutions. And based on the results so far, we think that it is the RNA structure that helps the pathogenicity of SARS-CoV-2 during infections, because because knockout virus, which does not have the ORF10 protein, did not, well, how can I say that? So when we try to produce the synonymous code changed virus, well, it produces the same ORF10 protein, but does not have the exact RNA structure. This is lethal to the virus.
Okay.
So we try to exclude the possibility that it is the protein by designing different reverse genetic and recombinant viruses.
Great. 1 more question.
Thanks, Ed. How does all the position of ORF10 compare to porcine coronavirus that's got ORF7? So ORF7 in TGV sits between 3' end, and the mRNA, even though you'd be higher. So I guess what other coronaviruses have been? And do we know what they do?
No.
Nice and easy.
Because I think most of the virology work focuses on the structural proteins and not a lot of work focuses on the accessory proteins, and especially those at the end of the 3' end.
Okay, thank you very much.